Welcome to Chapter 12, which is all about non-Mendelian inheritance patterns. The idea that not everything in terms of phenotypic and genotypic expression and inheritance is related to a simple monohybrid cross. So you should feel pretty comfortable with the idea that two possibilities within meiosis drive the recombination of maternal and paternal alleles when we see the phenotypic expression of offspring. And that would be the law of segregation, the idea that we're going to be separating maternal and paternal alleles, which can be either dominant or recessive, as well as the law of independent assortment, which is the idea that we might not just have gametes that are completely maternal or paternal with completely dominant or recessive alleles, which means that we should be getting from a purebred homozygous and a purebred homozygous recessive individual about 1/4 of alleles for either completely dominant, completely recessive, or one or the other, which should give us a phenotypic expression of 9-3-3-1 if we do a dihybrid cross. Now, starting in the 20th century, we started to notice that there were characteristics that didn't follow this sort of predictive pattern. And one was Morgan, who was taking a look at fruit flies, which was an amazing model organism to be studying because of a variety of things. They produced many offspring in a few weeks, so it was very easy to track generational turnover with phenotypes and genotypes. And there's only 4 chromosomes, which means it was very easy to track changes in alleles and genes and consider where those changes were occurring. And he noted that wild type, or what we would call normal phenotypes, were pretty common in the fly populations, but there were some mutant phenotypes, including white eyes, that were kind of prevalent and were atypical to the normal wild type red eye color. And here's a really good example of a zoomed in version of the compound eye of a wild or normal red-eyed fly, and here's the mutant or phenotypically atypical white eye. And in an experiment, Morgan mated male flies with the white eyes, which were mutant, with female flies with the red eyes. And what he noticed was that the F1 generation had all red eyes. And then the F2 generation showed the classic 3-to-1 red to white ratio. But what was interesting was that males only had the white eyes. So you would expect from a F1 generation cross of heterozygotes to have 1/4 be completely dominant, 1/2 being heterozygously dominant, and then the other 1/4 being recessive, but all of the recessive phenotypes were male. And he concluded that eye color must be related to the sex of the fly. And here's a good example of what that illustration or that experiment found. And what he concluded was that there must be a gene on the X chromosome of males, here's the Y chromosome of males, and if you inherited the recessive allele as a male, it must mean that you express it, while a female, maybe the recessive allele of red eyes would be masked by the red eye phenotype. And this is true for most biological organisms where there's a clear delineation between X and Y or sex-linked chromosomes. They're not always X and Y, but in males and females and humans, females tend to be XX and males tend to be XY. And we call any gene that's located in either of these sex chromosomes a sex-linked gene. There are a few Y chromosomal linked genes, but there are very few of them. Surprisingly, most of the biological female traits that are expressed in humans are found on somatic chromosomes, and the gene on the X chromosome that we find in males are often called X-linked genes. And X chromosomes have genes for many characteristics unrelated to sex. And I totally just said the completely wrong thing in the last slide. So X chromosomes do have genes that are not typical for sex, where Y chromosomes do encode genes related to sex determination. I am sorry about that mix-up. even teachers get it wrong sometimes. And I should know this because color blindness is a trait that we find on X chromosomes. Some disorders that are caused by a set recessive alleles on these X chromosomes, so color blindness, muscle dystrophy, and hemophilia, which is the ability to not have blood clotting, and all of these traits are highly more representative in terms of phenotypic expression in biological males as opposed to biological females, because females inherit 2 copies of any X chromosomal allele. In females, though, one of those 2 X chromosomes during fetal development and mitosis after birth are going to be called inactive Barr bodies, and basically they are methylated, which is a chemical that interacts with the histone proteins on DNA to cause them to condense, and that is a random, which can sometimes lead to some very interesting phenotypic expressions within things like fur color within cats, where heterozygotes tend to be different for each particular cell that expressed either the dominant or recessive allele. And we can see that here where in some cells during fetal development, the active X codes for black fur, and then in other females, they repressed that X chromosome, so the X chromosome that's now activated codes for an orange color. This can also be seen as heterozygously incomplete dominance, where we might see a phenotypic expression in females where both are being activated or expressed, which means that you might not have cystic fibrosis, but you are a carrier, so to speak. Not that cystic fibrosis is linked on the X chromosome, but the same idea of showing sort of incomplete dominance where you're not quite homozygous recessive or dominant with the trait expression. And this is partially related to the notion that genes are linked on chromosomes, and sometimes during crossing over in synapse and meiosis, we can have some recombinations. So linked genes are genes that tend to be inherited together because they're on the same chromosome. It's sort of like ordering food at a restaurant, and there are certain foods that come on certain types of meals. You might love pickles, but you can only get them on a hamburger, so to speak, at a restaurant. So ordering the chicken sandwich might not be the best choice if you're looking to get pickles. And how does linkage affect inheritance? Well, Morgan did experiments with fruit flies that showed that linkage affects the inheritance of 2 characters, and he did that by crossing flies that differed in traits of body color and wing size. So he was examining dihybrid females, so meaning that he had females that had both one copy of the wild and mutant alleles for eye color and wing size, and he crossed them with homozygous pure breeding recessive males in a test cross, and most offspring tended to show either complete homozygous recessive traits or heterozygous dominant traits. So the test cross showed that when we took a female that was homozygous for both gray bodies and normal wings, and a double mutant black body, we tended to get dihybrid females and recessive males. So when we look at this, we can really think about how alleles are being donated by different types of gametes. So this goes back to the sex-linked traits are inherited based on your chromosomal inheritance pattern. So when we're thinking about the test cross of these 2 individuals, this female that's a heterozygous can donate either a wild type or mutant allele for eye color, and for wing type, well, really there's only one option for the homozygous recessive sperm coming from this mutant male, which is why there's only one row for the sperm, but there's 4 different possibilities for the eggs, and that gave us sort of a 1 to 1 to 1 to 1 ratio, or we would expect. But what we found actually when it came to the actual results is that we found these two phenotypic expressions were much more common than being a hybrid of the mutant and the wild type traits. So just let's reconsider that a little bit more. We're doing a cross of a heterozygous female fly with a homozygous recessive male, which means that we really should have a 1 to 1 to 1 to 1 ratio based on the expression of these traits. But we're not getting that. Something must be happening. And what was happening is that these alleles are linked, so they are expressing recombinant chromosomes. So instead of having what we were expecting to see in terms of just either purely heterozygous or purely, or not purely heterozygous, purely homozygous alleles, we're sort of getting these hybrid recombinant chromosomes that are maybe changing the expression of our traits. So when we looked at the actual recombinant chromosomes of not being just purely one or the other, we got about 17% or 391 recombinants out of 2,300 total offspring, or about a recombinant rate of 17%. And what this helps us understand is that basically the further apart two genes are, the higher the probability that a crossover is going to occur between them, and therefore they will have a higher recombination frequency. So this rate of 17% isn't that large. When we think about what's on a linkage map, the further away two alleles are, the more likely they are going to cross over during some sort of meiotic division. And a gene map or linkage map basically shows us the frequency of that probability of genes being recombined, where 1% is a unit showing that there's a 1% rate of recombination. So there's about a 9% chance that B, which is eye color, and CN, which I believe is body color, are overlapped. And there's a 9.5% chance that this allele and this allele are going to be crossed over. But if there is a recombination rate, there's a higher rate that all of these are going to be combined. And we can basically see how those alleles are linked on any organism in terms of their rate of recombination. Now, there are some other aspects of non-Mendelian genetics that are important to cover. The first is non-disjunction, which is where meiotic division is abnormal. And this is because there might be a problem with disjunction of the homologous chromosomes or the sister chromatids during meiosis 1 or meiosis 2. And that generally results in either some normal and some abnormal, or all abnormal chromosomal inheritance in gametes. And this is also called aneuploidy. We can be monosomic or trisomic depending on the chromosomal copies, 1 or 3. And polyploidy is where an organism actually has multiple sets of chromosomes, maybe 3N or 4N. This is pretty common in plants that have been artificially selected for by humans to increase things like starch production or sugar production. Now, there are some structural problems with chromosomes. We can have deletion, duplication, inversion, or translocation, and all of these tend to either modify a gene's expression, the ability to express that gene, or the phenotype of an individual. And then we can also have really specific types of inheritance patterns based on those problems. So trisomy 21 is Down syndrome, where an individual receives a third copy of chromosome 21, and that generally is related to an increase in the age of mother at birth, and that can have some pretty phenotypically strong changes in terms of the person's characteristics. They tend to have some learning cognitive problems. They tend to be sterile. They can live long, happy lives, but there's obviously some pretty strong phenotypic changes from a quote-unquote normal human being to them. And then you can have non-disjunction of sex chromosomes. So Klinefelter syndrome is where males tend to be biologically sterile with some aspects of female sex characteristics when they inherit a second X chromosome. And females can be either trisomic X, which means they're just slightly taller, or monosomic X, where they tend to be sterile. And these are the only known viable, this is the only known viable form of monosomy in humans, because any karyotypic expression with any somatic chromosome with just one copy would mean that mitotic division would not be possible. Another disorder caused by structurally altered chromosomes is Cry-du-Chat, which is a specific deletion of chromosome 5, and this person tends to have a cat-like cry in terms of its vocal expression. And you can have things like chronic myelogenous leukemia, which is caused by translocation of chromosomes, where chromosomes 9 and 22 have translocation of those genes. So in a nutshell, we now know that we can have types of inheritance that are not typical of Mendel's traits based on a variety of alterations within chromosomes. Let me know if you have any questions. I'm sure the gene-linked map will be something that we talk more about in class that is a bit confusing.